Hello everyone, welcome back. In this session, we are going to learn how electrical signals are transmitted in a single neuron and between two neurons. Upon completion of this video, you will be able to learn the following: the definition of the cell membrane and the resting potential. On the top of that, you will learn a very important concept called action potential. You need to understand its all or none principle and the 7 key phases of action potential. You will also learn the detailed structures of synapses, synaptic transmission, and 4 types of synaptic connections. Finally, I will show the key components for neural signal processing. Having learned the basic structure and different types of neurons, we are now ready to study how signals travel down an axon. More importantly, we should pay attention to how the voltage of a neuron changes during the signal propagation. This is all about action potential. Before detailing the action potential, I will first introduce some basic notions. The first concept is membrane. Let's look at this neuron. We zoom in the area of its axon. We notice the yellow nerve fiber. When magnifying this region further, we can observe the cell membrane of this fiber, which is a double layer of lipids and proteins that separates the contents of the cell from the external environment. This region is the contents of the cell, which is inside the cell. And the opposite part is the external environment, which is outside the cell. It is important to note that there are different ions concentrated on each side of the cell membrane. For example, there is a high concentration of potassium ions inside the cell and a high concentration of sodium ions outside the cell. The differences in the concentrations of ions on opposite sides of the membrane lead to the cell having a voltage called the membrane potential. Using a voltmeter, we now place one electrode on the inside of the cell membrane and the other on the outside. We see the voltmeter reads -70 millivolts. This number indicates that the inside of the neuron is slightly negative relative to the outside. This is called membrane potential. The membrane potential is what we use to describe the difference in voltage, or electrical potential, between the inside and outside of a cell. Membrane potential is also known as membrane voltage, which is mainly caused by the unequal distribution of ions on different sides of the cell membrane. In this example, we can see the number of positive ions inside the cell is smaller than the outside. Therefore, the inside of a neuron is more negative than the outside. Scientists also use voltmeter to measure the voltage of many other neurons. Interestingly, they discovered that as long as neurons are at rest, the membrane potentials for all these neurons are quite close, being approximately -70 millivolts. We call this membrane potential the resting potential. Note that we say a neuron at rest, meaning that the neuron is not sending a signal. That is, the neuron doesn't fire. Formally, the resting potential is defined to be the difference in voltage across the cell membrane when a neuron is at rest. In short, the resting potential is the membrane potential for the resting neuron, which is approximately -70 millivolt. Here is a question: why a neuron at rest has a resting potential of -70 millivolt instead of 0? To answer this question, I will first introduce two types of channels: leaky channel and voltage-gated channel. Both types of channels allow ions flowing across the cell membrane. However, their gating mechanism, that is, what opens and closes the channel, is different. Leaky channels are always open, allowing free passages of sodium ions and potassium ions across the membrane. For example, potassium ions can easily pass through the leaky channel across the cell membrane. In contrast, the voltage-gated channels are only open at a certain voltages and are often closed at the resting potential. For instance, when the voltage is minus 70 mV, the voltage-gated channel is closed. When the membrane potential increases to a certain level, the channel is open, allowing potassium ions flowing across the cell membrane. When the membrane potential decreases to the resting potential, the channel is closed again. Based on leaky channels, we are now ready to explain why the resting potential is negative. There are two reasons. Firstly, there is a higher concentration of the potassium ions inside the cell. This creates a potassium ion gradient, enabling potassium ions to pass through the potassium leaky channels easily. Therefore, the inside of the cell becomes more negative. At the same time, there is also a concentration of sodium ions outside the cell. These sodium ions are coming in to the cell through sodium leaky channels. If this process continues, then more and more sodium ions are inside the cell, and potassium ions are outside, leading to an unbalanced distribution of ions on each side. To keep it balanced, there is a sodium-potassium pump that uses energy to move 3 sodium ions out of the neuron while bringing just 2 potassium ions in for each type. So, the net result of the pump is to take 1 positive ion out of the cell. Consequently, after some time, the number of positive ions inside the cell is less than the outside, which produces the resting potential. Next, we are going to study an important concept: action potential. An action potential is the rapid changes in voltage across the membrane. It occurs when a neuron sends information down an axon. An action potential begins at the axon hillock and reaches the axon terminal. An action potential is also known as a nerve impulse or a spike. It is worth noticing that not every stimulus would trigger an action potential. As we know, in the resting state, the membrane potential of a neuron is around minus 70 millivolts. An action potential is generated only when a stimulus changes the membrane potential to a certain threshold, say minus 55 millivolts. The value minus 55 millivolts is called a threshold potential. The generation of an action potential follows a principle named all or none. Any subthreshold stimulus will cause nothing, whereas threshold and superthreshold stimuli produce a full response of the excitable cell. For example, let's assume the resting potential of a membrane is minus 70 millivolts, and the threshold for the action potential is minus 55 millivolts. If the stimuli change the membrane potential to minus 65 millivolts, then an action potential will not occur, because it is below the threshold minus 55 millivolts. However, if the stimuli change the membrane potential to minus 50 millivolts, which is above the threshold, this will trigger the action potential. Again, if the next stimuli only slightly change the membrane potential to minus 60 millivolts, then the action potential will not occur, as it hasn't passed the threshold minus 55 millivolts. That is all or none principle. This principle happens at axon hillock, where the action potential begins. We can imagine this is the axon hillock. If there are only a small number of ions reaching the axon hillock, they are unable to pass through, as they below the threshold minus 55 millivolts. However, with the increasing number of ions reaching the axon hillock, they can pass through the axon hillock as they reach the threshold. In this case, the action potential is triggered. An action potential consists of several phases. Before detailing each phase, I will first introduce three terminologies. The first terminology is polarization. A neuron is said to be polarized if the outside of the membrane is positive and the inside of the membrane is negative. For example, the resting potential describes the unstimulated, polarized state of a neuron. If the membrane potential becomes more negative than it is at the resting potential, then the neuron is said to be hyperpolarized. For example, this neuron is hyperpolarized since its membrane potential is minus 80 millivolts, less than the resting potential, minus 70 millivolts. If the membrane potential becomes more positive than it is at the resting potential, then the neuron is depolarized. For example, this neuron is depolarized as its membrane potential is positive 40 millivolts, greater than the resting potential, minus 70 millivolts. Next, we are going to learn how an action potential is transmitted. Let's look at this picture. This is the cell membrane with three voltage-gated channels. Two orange channels are sodium channels, and the one in blue is the potassium channel. The green one is a sodium-potassium pump. Firstly, at resting potential, the voltage-gated sodium and potassium channels are all closed. Then, the stimuli, such as light and sound, cause some voltage-gated sodium channels to open. Once we get to the threshold of minus 55 millivolts, the action potential begins, with many other voltage-gated sodium channels opening. With the sodium channels opening, depolarization occurs. Sodium ions rapidly rush into the cell, and the voltage rockets up to positive 30 millivolts. At this point, voltage-gated sodium channels closed. Potassium channels now open, and repolarization occurs, as potassium ions rapidly rushed out of the cell. Then the voltage goes down and overshoots minus 70 millivolts before the potassium channels can close. Finally, the sodium and potassium pump restores the resting potential. 3 sodium ions are moved from the inside to the outside of the cell, and 2 potassium ions are brought into the cell. That is the propagation of an action potential. Next, let's move on to the last concept, neural synapses. A synapse is a junction between the nerve cell and its target tissue. Here, The target tissue can be another nerve cell or a muscle cell. Most synapses are chemical. The action potential is transmitted from the axon terminal to the target tissue by the chemical substances called neurotransmitters. In this animation, we can see that there are many yellow chemical substances released by a neuron. They are neurotransmitters. There are two types of neurotransmitters, excitatory and inhibitory. If a neurotransmitter stimulates the target cell to an action, it is an excitatory neurotransmitter. In contrast, if it inhibits the target cell, it is an inhibitory neurotransmitter. Next, we are going to look at the details on synaptic transmission. Most communications between neurons occur at a synapse. Let's now zoom in the synapse to see its structure in detail. This is the presynaptic neuron, which transmits the signal towards the synapse. This is a postsynaptic neuron, which transmits the signal away from the synapse. We can see that the neurons are not actually connected to each other. There is a very small gap between the presynaptic and postsynaptic neurons. This small space is called the synaptic cleft. The cleft is less than 40 nanometers wide. In comparison, a human hair is about 75,000 nanometers wide. In the presynaptic neuron, there are chemical signals called neurotransmitters that are packaged into small sacs called vesicles. In this picture, the green balls denote vesicles. This is the receptor on the postsynaptic neuron. Each vesicle may contain thousands of neurotransmitter molecules. When the presynaptic neuron is excited by an action potential, this causes the vesicles to fuse with the presynaptic membrane and release their neurotransmitters into the synaptic cleft. Once in the synaptic cleft, the neurotransmitters interact with receptors on the postsynaptic membrane. They bind to the receptors and can cause an action to occur in the postsynaptic cell. This action may increase the chance to get the postsynaptic cell activated and fire an action potential. This is how the signals are transmitted from one neuron to another through neurotransmitter molecules in the synapses. Last but not least, let's take a look at the 4 types of neurosynaptic connections, which depends on the location where the synapses connect to the postsynaptic neuron. Axodendritic synapses refer to the axon terminal connecting to a dendrite, while axosomatic synapses refer to the axon terminal linking to the cell body. These are the 2 most common situations. There are also 2 cases. One is axo-axonal synapses, which refers to the axon terminal connecting to the axon. The other is dendrodendritic synapses, which refers to the link from dendrite to dendrite. Now we have learned the basic structure of a neuron and how electric signals are transmitted within a single neuron and between 2 neurons through neurotransmitters. To summarize, let's take a look at the following key components for neural signal processing. Firstly, signals from neurons are collected by the dendrites. Then, the soma aggregates the incoming signals. When sufficient input is received, that is, a threshold is exceeded, then the neuron generates an action potential. The action potential is then transmitted along the axon to other neurons or to structures outside the nervous systems, such as muscles. If sufficient input is not received, that is, the threshold is not met, then the inputs quickly decay and no action potential is generated. Timing is also very important. Input signals must arrive together. Strong inputs will generate more action potentials per unit time.